I'm Meredith Blackwell, and this is Rob Samson, and we're at IMC 11 in San Juan, Puerto Rico, and we are doing an oral history of mycology. And our first interview is with each other, and so that will be sort of a test for the system that we're using. Our videographer, Dustin Howard, is all ready to go. So what I'm going to do is say that Rob and I have known each other since 1976. We met in Gainesville, Florida. But I want Rob to tell me about his life before that momentous event. Where were you born?
I was born in Indonesia, in Bandung, in the island Java. And my parents were in fact Dutch. In fact, my father was Indonesian German, and my mother was Dutch Chinese. Since they had the Dutch nationality, they had to go out of the country in 1950 because Indonesia became independent. So that was a very dramatic leave, because we had to go by boat from Indonesia to Holland. And just imagine, they couldn't prepare that trip, and we arrived in Amsterdam, in fact, during the winter.
What year?
That was in 1950, September 1950. My father was in fact in the army, the Dutch army in Indonesia, and he became another position in the Air Force in Holland. And we moved to the southern part of Holland. And we lived between the farmers in a very small village.
Near Maastricht?
No, it was near Breda.
Okay.
And which was really interesting, because we were the first, let's say, people they have ever seen from another country. And I remember the first day when we were in the house, all people came out and had a watch how these Chinese Indonesian look like. It was very strange. But we grew up there, and I had a very good time there. I went to school there, and then after that I went to university, first in Eindhoven to do chemical study, but I found that biology was much better. And I moved to Utrecht, and in Utrecht I did my master, my bachelor, my master, and after my master, which one part was in the on fungi, was just by accident I came to the CBS, the Central Bureau of Schimcultures, which is now called the Westerdijk Institute. And I had just one project to go, and I went to Barn, and I was very lucky, because at that time Dr. van Arx, the director, just got a new building from the academy, and I could start, and I started with Cunningham Mella, and I had a great time. I was completely fascinated by the fungi, and when I finished my project in 6 months, he said, "Whoa, this is good, we're going to publish this." That was in 1969, my first publication.
So were you interested in fungi before?
No.
Just somebody said, do this?
Yeah, and I was probably interested in the environment, but also I was really fascinated by the microscope and by the beautiness of the fungi. And then in 1969, I finished my master, and then he said, "Rob, do you want the job?" Okay, so he gave me a job, and I said, "Okay, where do I sit?" "Just pick a room. We have still 4 left and empty." So since then I was at CBS, and now I'm working at the Westerdijk Institute, which is the name changed in 2017. In the meantime, we moved to Utrecht, and at this time I worked 48 years for this institute.
Woo! So you never really left?
I never left, but I did several sabbaticals. I did my postdoc. That's where we met.
Yes,
I did my
in
postdoc
Gainesville with Kimbrough.
Yeah, I did my postdoc with Gary Cole in Austin.
Oh. Okay.
And it was a very fascinating time. I think the best time to be educated as a mycologist.
It's funny, because I went to school at Texas, and I never realized till today
That we are the same.
Yeah. And then you followed me to Gainesville.
Well, I didn't follow you. I didn't know you. So I got a position or a project with on intermogenous fungi, Nomoria, And there we met. I got a position by Jim Kimbrough, and you were there.
That's
you
where
worked with Henry Aldrich. Yeah.
That's right. And then I was actually in Kimbrough's lab
Okay.
by then.
Yeah.
Excuse me. Well, I was born on the bayou, what we called the small rivers of Louisiana, South Louisiana. And so I was in a place right near the bayou, and we saw poisonous snakes, all kinds of oak trees, poison ivy. So by the time my father returned home from the war, when he was in the Netherlands, that's why I said Maastricht, because he really liked Maastricht, and then also in Germany. So he comes home and he began to teach me the poisonous snakes, because we had lots of them, and poison ivy and those sorts of things. But in school I was never interested in biology because I couldn't spell. And so even in college I majored in things that would allow me to take math instead of biology. So I went, by that time we lived in Lafayette, Louisiana, and so I went to the local college there. And then I went to the University of Alabama because I had decided by that time, after going deep sea fishing, that I really wanted to be a marine biologist. So at Alabama, I got my master's in ichthyology. And so I went to Texas. I had intended to go to Tulane and work with the ichthyologists there. But what happened was that I got married instead. And so I had to go to Texas, where my husband was going. And I didn't like the ichthyologist there. We didn't get along very well. He wouldn't take me on the field trips. So I quit and started looking for a job. And by that time I had a couple-month-old baby, my daughter Elise. And it was very, very difficult for me to find a job. But Henry Aldrich, who had worked for Dr. Elix on his grant, Henry got a fellowship, so he didn't have to do any work except his own. So Dr. Elix hired me rather reluctantly, but it was a wonderful, wonderful time. And one of the first fungi I found was one that was known only from Java.
Oh, really?
It was a slime mole, actually, not a fungus.
Yeah.
So it was interesting. And it was also there that I isolated a fungus from dirt right in the back next to the building in a flower bed, dry habitat. And I was supposed to get Cunninghamella. That's what everybody in the class said. Oh, you can isolate Cunninghamella back there. No Cunninghamella. But instead I got this thing that was strange, couldn't figure out what it was. Dr. Elix had never seen it. He thought it might be insect eggs. He said, go try to grow it anyway. And we grew it out, and it turned out to be something that was called Echonosporangium, that David Malik described.
Oh, yes.
Right.
Very rare, in fact.
Yeah, well, it had been found 3 times in a 2-year period.
Yeah.
Ranzoni in the Sonoran Desert, Dave in, I think it was Nevada, in a desert area, and the Texas Botany Building. So it was very, very strange. No one's ever found it again, and I've been looking through all of these sequences. People do the environmental sequences. It's not there.
So you worked, or you were also in Austin, and you worked with Alexopoulos, Dr. Alexopoulos. How was he? Because when I was in Austin, he was in this very small, very dark room. He was, in my opinion, very shy, never came out. But you worked with him. So how was your experience with him?
Oh gosh, he was wonderful. For the first couple of months I worked for him, I was Mrs. Blackwell. And I can remember how happy I was the day he called me Meredith, and he finally did after a couple of months. But it was when I found this slime mold and some material he had in moist chamber from Mexico, and it was the one from Java. It was called it's a different genus. It's Spicyrina is the genus name. I had forgotten for a minute. So after I found that, and he saw it, and it was right, because I'd looked through all the picture books of slime molds, and it was very distinctive. So I was in with that. But when you saw him in 70, when would it have been?
75, 76, yeah.
Yeah, by that time he had had several benign brain tumors, and he saw double vision. before that, he had been diagnosed with myasthenia gravis, but it turned out all the time it was the brain tumor. So he was really debilitated by that.
Yeah.
And he was totally different from the early days when I was there with him.
Yeah. Because when I met him, he asked me what I was going to do in Austin. I said, well, I'm going to study with Gary Cole on the corneogenesis of hyphomycete. And he said, well, this is really a very nice study. But that was the only conversation I had with him, which is a pity because he was an extremely famous man.
Oh, yeah, and in those days, though, he just had been so ill.
Yeah. Well, why are you not proceeding your study on myxomycetes? Because you were really very specialized in that field, isn't it?
Well, the problem with myxomycetes is there are fewer than 1,000. I was just more interested in other things. I was interested in insects and the things that were associated with them. And so I guess the first things I worked with after that were with Kimbrough. In his lab, some of the little ectoparasites of termites that had been discovered by Thaxter, Roland Thaxter, and never seen until they began to find
I just remembered, didn't we have a joint
we had a paper? We did.
I forgot about that.
We had Brazilian termite fungi.
Yeah, that was published in Mycotaxon, isn't it? In 78, 79, something.
I can't remember where it is. But
I forgot the name, so it's a long time ago. Yeah.
I don't remember. Yeah. Oh, it started with an E, but that's all I remember.
So you did also electron microscopy.
That's right.
And particularly on Ascomycete spores, isn't it? Ascospores.
Well, actually, the first was with the slime molds. Dr. Alex believed we should learn to use the latest techniques, and at the time, TEM was the technique. And so, that would be one thing. He gave us such good advice. He says, I don't know how to do that, but you learn, and then you keep up with the latest technique always. Yeah. And so I think later we will interview John Taylor, and that's one thing I want to ask him about. As an electron microscopist, he was very much converted with the DNA studies he's done.
Yeah, I learned quite a lot of scanning electron microscopy from Gary Cole, and when I came home from my postdoc, my director said, what did you do? And I said, I did scanning electron microscopy. And he said, do you want one? I said, okay.
No.
Yeah, he was, at that time I think money was no issue. So we bought a scanning microscope, and nowadays I have no time, but there are other colleagues in my institute who are doing this. But what you see in a scanning microscope is really wonderful, and you're again fascinated by the beautiness of the fungi.
They're very beautiful. And the problem with it is, though, you learn so much more different things from the transmission. I always like that better.
Yeah.
And it was nice this morning in one of the talks I saw TEM.
Yeah.
And it had been used to get information, so that was good.
Yeah, there were congresses where everything was TEM. Now you see only trees.
I remember when I worked with Henry Aldrich for 1 year, he would go to meetings just armed with a pile of electron micrographs, and all the EM people would get in the corner and show each other pictures.
Yeah. In fact, what we also have common, I just think about it, is the insects and the fungi. Because particularly in the 70s, I worked quite a lot with Harry Evans on entomopathogens, and we described quite a lot of papers with new
species. And you wrote a book, but it wasn't with Harry.
It
was with Harry. Yeah, because I remember I sent you some plates.
And I just asked for do a second edition. Let's see if we have time. So I think this is one thing we have in common. Also that the interrelationship between fungi and insects, and you have done also quite a lot of insect fungus related papers, isn't it?
Yeah. And then I was just going to tell you, actually lately I've worked on aspergillus. Could you believe that?
Oh gosh, nice.
I was out in Arizona. I worked for a time collecting with Bob Gillertson at the University of Arizona, and Gil was very interested in slime moles out there, and the desert is full of slime moles. But we were in one of the riverine valleys, and we were looking at, we were sitting down looking through leaves for slime moles, because there's some that inhabit leaves. And all of a sudden we found an area that was probably 10 by 20 feet, and the oak leaves were just covered with what looked like a slime mold. And we started looking at it, and it didn't quite look like a slime mold, but we thought, well, we better collect it anyway. And it turned out to be a Cinematous penicillium.
Wow.
And so I've looked at it. Unfortunately, Keith Seifert beat us to it with some Spaniards who also found the same thing.
Yeah, this is the Talomite. Yeah, Cidicula. Cidicula.
Yeah.
Yes, yeah.
And so then, though, there is a sequence of it from Arizona in Betsy Arnold's herbarium, and lo and behold, it's the same thing. Because last year, after 40 years, we were able to get DNA from it, and one of Kathy Ain's students was able to sequence it for us.
Yeah. So regarding the insects and associated fungi and particularly yeast, you made some estimation about the number of fungi in nature. Do you think that particularly the insects are an enormous, let's say, pool or a source of unknown species?
We're still finding new yeast so easily. I don't know for other fungi, but the yeast in the gut, they're just tremendous numbers. We've looked in Thailand, Australia, Panama, Venezuela, and then the U.S., and we keep finding new species. So I don't know. But actually, the estimate of 5.1 species of fungi, 5.1 million species of fungi, that was actually based on work that O'Brien had done in Rita's lab, Rita Vilgelis's lab, and that was based on soil fungi.
Yeah,
so,
of course.
You know, it depends on what you look at. Kathy Ain was working in a place in Guyana, I think it was, and if you had looked at that where there was one tree, dominant tree, and they collected, 100 species, well, then you would have a very different kind of ratio there.
Yeah. In your career, you educated quite a lot of students, which are becoming already now very famous. Also, what is your, let's say, approach when you are going to educate a student in mycology?
Well, first of all, I should say I really have had relatively few students, because I always felt that instead of coming to LSU, unless there was a real reason to be there, they should go where there might be more mycologists. But anyway, my way of doing it was what I learned from Dr. Alex. You let people pick a project of their own, something they're interested in, but still within your area. Yeah.
Well, my thing is that they should look through a microscope. And nowadays it's sometimes a bit disappointing that biology students come and they say, I like to do mycology, but I don't like the microscope. I said, well, then you don't have a project with me, because I believe that the biology of a fungus and the morphology of a fungus is equally important as the sequence. And I've seen so many mistakes with biologists only doing sequences and never look to a fungus.
Exactly.
And this is not the way we do. However, I fear that the trend is that the molecular methods are going to win from morphology, even with identification. And I see this particularly in applied fields like food mycology or indoor mycology, that people don't want to look in microscopes and try to identify at least the genus, They just want to do.
Well, the problem is your fungi are so hard to identify.
Well, but they're important.
Yeah, no, I agree. And with yeast, though, it's, we have like 100 traits we have to look at in a sheet. So there was never any question that the students would look at the yeast. And we I wasn't doing electron microscopy at that time.
Yeah.
But no, you're right. And I think you avoid making mistakes, because I know several people who have mixed up DNA in the early days. And in fact, when I did the textbook with Charles Mims, there were several problems with that. And if the people had only looked at what they were doing and looking at the specimens as well as the
but
DNA.
still today, you know that several genomes are not the species they were originally sequencing. This is really a rather serious mistake. And this is just due. They have never looked to the strain and so on. Yeah.
Yep.
But are there other, let's say, tips to make a student really enthusiastic for mycology? Or is this just in the student itself, when he's fascinated by the fungus, by the beautiness of the fungi, and of course if the project is interesting?
Yeah, I think that if they're interested in mycology, it's usually because they are introduced to it. Many people are never introduced to it. I was talking to a young man called Tim who came here from Kiev on my flight. I was sitting next to him, and he said in Kiev they had to each take a mycology course in their first year of study. And then there was a follow-up where they did a field study, a field camp with mycology.
Yeah.
So that's a way to make sure people know, because I think we need more mycologists. You know, plant taxonomists, they do different kinds of projects. They can do more fine-grained stuff. But mycologists, we need mycologists to do more basic.
But is it not difficult? Because I do not see it only in the US, but also in Europe, that a lot of mycological institutions are just closed because somebody died, passed away, and there is no successor. So there is not a mycology in the curriculum of the let's say, education of the of students. So we are losing quite a lot of mycology. I'm fortunate that I'm working at the Westerdijk Institute because
that's a wonderful place.
We are in a centrum where everything is happening, and we are really well supported by the Academy of the Netherlands Academy. So we are lucky, but I see other institutions where, you know, somebody is retired and then there's no mycology left. And
well,
I think that
Oberwinkler retired, they didn't hire a mycologist, did they?
No. And that's also in other German institutes or Belgian. You just
when I retired, they didn't hire anyone in mycology.
Well, that's something very sad, in fact. Yeah. But I don't know how to deal with it, with this problem. That
when there are 5 or 6 vertebrate taxonomist and one mycologist, it doesn't make sense if you look at numbers of organisms.
Yeah. Let me ask you something else, because you were in international commissions and so on. How do you think that this is important for mycology students to be involved in not only science but also what I also call a little bit politics? How to let's say make mycology more prominent in the microbiology or even in the botany. Now, I myself, I'm the Secretary General of IUMS, the International Union of Microbiological Society, and I work together with virologists and bacteriologists, and we have quite an active division on mycology, which is now also using algae and so on, so eukaryotic microbiology.
That's how I got to Prague.
Yeah. So this I think is important for students, that it's not only a fungus. The fungus is not by himself in nature. This is more broad. It is really microbiology. And
did you attend our session last night?
No, I didn't.
That was the topic. You know, it was fungi, and inside fungi were bacteria, and inside bacteria were viruses, and on and on and on. And it's like Jonathan Swift's little poem about big fleas have little fleas and all that. I don't remember exactly. But the other thing is, there's so many times people think that a fungus might be causing something, and I think of the grass at Yellowstone National Park in the United States. They thought because it was infected with a fungus, an endophyte, it was able to withstand high temperature. And then someone comes along and finds out, well, it's really the fungus that's infected with a virus. And that's the ones that are able to withstand. And then there are a number of cases with some of the insect-associated things, like the plant hoppers. Those are really interesting stories with the genomes now showing there are all kinds of different bacteria and maybe viruses.
Yeah.
So it's very, very interesting. And so you're exactly right. People need to be a little bit broader and keep their eyes open for this kind of thing.
Yeah.
But our speaker last night was very good at pointing out the work they had done. So, yeah.
Okay.
Now, what are you going to do? You retired. I retired. What are you
Well, I
doing?
retired already in 2011. Officially in Holland you have to go when you are 65, but I got another assignment for 5 years. But in this assignment, this period, I tried to educate or raise my successor, Jos Houbraken. He's a very good scientist too. So now my assistant is my boss, and I have to ask him, can I go to Puerto Rico? And he said yes, of course.
Oh, did he come?
No, he doesn't come. He stayed at home. But yeah, I still like to work on mycology. It's it's my second wife, I always say, and it's very nice that I by accident came into mycology and had a chance to work in an institute like the Westerdijk Institute, and also had a chance to travel a lot and to see a lot of people, visit the laboratories. Sometimes you see, oh, they're better, but we have this and this and this. And the network, I think, is extremely important. So if you go as a student to a conference, I think you should really not be shy and talk to the people. Because most mycologists are really very nice people, isn't it?
Yeah, especially if a student comes up and says, oh, you're my hero. This happened to me yesterday. So we're always happy to talk to any student. So. And I just met this morning a young man from Brazil that I talked to in Amsterdam last, who's working with Escovopsis. So it was really interesting to find out what he's
is this
done.
guy there? Because Escovopsis I saw for the first time.
Andre, the one from Brazil,
yeah,
Andre Rodriguez.
I have to speak with him because I know that we couldn't identify that species, and I had to read very old German stuff by Müller. And I remember that I took the book on vacation. I read the whole German book, and I saw that this is the species what we're finding in these termites. Yeah, it's a really, really interesting story.
The ants.
Yeah, ants. Yeah. And there are many species. So
that's right. And he said he's found some new genera too.
Yeah. Very interesting. Yeah.
So we learn from talking to students.
Yeah,
well, he's no longer a student, but he's a young mycologist.
Okay.
Yeah. Well, this has been fun.
Yeah,
and
very fun.
I think we probably should finish up, and we look forward to interviewing people for the rest of the week.
Thank you, Meredith.
Thank you.